Hello, welcome to the Selfie YouTube channel. Today I'm going to be reviewing everything you need to know for Unit 5 on the AP Biology exam. I plan to make more videos for the other units, so check those out when I post them. Also check out the Selfie website for my notes on each lesson of Unit 5. The link is going to be in the description. So meiosis is a type of cellular division performed to create gametes or sex cells for sexual reproduction. To get a better understanding of the cells involved in meiosis, you need to understand that a diploid cell is a cell with two full sets of chromosomes, one set from each parent. It is represented by 2N. In humans, the total number of chromosomes in these cells is 46, so 2N would equal 46. Some examples of diploid cells are body cells like skin cells and leaf cells. A haploid cell is a cell with one set of chromosomes. It is represented by N. In humans, the total number of chromosomes in these cells is 23, so N would equal 23. Some examples of haploid cells are gametes or sex cells, which is eggs, sperm, and pollen. When two haploid gametes, like an egg and a sperm, come together in sexual reproduction, a diploid cell is created. In meiosis, a diploid parent cell produces 4 haploid daughter cells, sex cells. Okay, so let's get into the stages of meiosis. There are 2 parts of meiosis, meiosis 1, where the parent cell divides, and meiosis 2, where the daughter cell divides. Okay, so in meiosis 1, prophase 1, the DNA coils into chromosomes. The nuclear envelope begins to disappear, and the fibers that pull the chromosomes apart form. It's important to note that the chromosomes are now double chromosomes. Non-sister chromatids exchange genetic info, also called crossing over. In metaphase 1, chromosomes remain in pairs, still tetrads. Fibers align the pairs in the center of the cell. And anaphase 1, fibers separate the chromosome pairs. The sister chromatids are still attached, so please note that, and move away from the center. So anaphase, away, they both start with A, that's a way to remember. Telophase 1, nuclear envelope reappears. Cytokinesis occurs, which is the division of the cytoplasm, and creates 2 daughter cells. Meiosis 2 prophase 2, the nuclear envelope begins to disappear and the fibers form again. Metaphase 2, fibers align the double chromosomes in the center in a single file line. Anaphase 2, fibers align sister chromatids and pull them away from the center. The chromosomes are now single chromosomes. Telophase 2, the nuclear envelope reappears and the chromosomes uncoil. And of course, cytokinesis occurs again, which is just the division of the cytoplasm. So now the 2 daughter cells from meiosis 1 are now 4 daughter cells. Okay, now that we got that down, make sure you don't confuse meiosis with mitosis. Mitosis produces 2 diploid daughter cells, or clones, that are genetically identical to the parent. Meiosis produces 4 haploid daughter cells that are genetically different from the parent and each other. Because the haploid daughter cells are genetically different, meiosis generates genetic diversity, which means it makes it possible for the offspring to inherit a wide range of characteristics from the parent. Let's analyze the processes that increase genetic diversity in meiosis. So the crossing over of non-sister chromosomes happens in prophase 1 of meiosis 1 and results in recombinant chromatids. Another process is the random assortment of chromosomes in metaphase 1, so it causes the crossing over to vary. And fertilization also increases genetic diversity because it takes genetic information from each parent and it is considered random because any gamete can contribute to the offspring's genome, which makes genetically different offspring. Okay, so before I go into Mendelian genetics, these are some important vocabulary words to write down in your notebook. So number 1 would be gene, which is a unit of coding for traits passed down from parent to offspring. Trait, genetically determined characteristic of an organism. An allele, which is different versions of a trait. An example is blue eyes or brown eyes. It's inherited from both parents. Different alleles can be inherited for the same gene. There are two types of alleles, dominant alleles and recessive alleles. Dominant alleles always show up in the phenotype if inherited, and it is represented by an uppercase letter. A recessive allele only shows in the phenotype when the dominant allele has not been inherited, and is represented by a lowercase letter. A genotype is a combination of inherited alleles and is usually represented by two letters. And there are two types of genotypes, which is homozygous. Homo means same, and the genotype, this is a genotype containing two of the same alleles. An example is capital A, and lowercase a, lowercase a. Heterozygous, hetero means different, is a genotype containing two different alleles. An example is capital A, lowercase a. Another vocab term is phenotype, which is a physical result of the genotype, and it is what can be observed. So if someone is tall or short, monohybrid cross is an examination of how one trait is inherited, and it is observed through a Punnett square. A dihybrid cross is an examination of how two traits are inherited, also done through a Punnett square. And autosomal is relating to the body. So autosomal chromosomes code for everything in the body except for sex. Okay, now that we're done with vocabulary, let's get into DNA and RNA. So DNA and RNA are both nucleotides, as you should know from Unit 1, and make it possible for genes to be transmitted from parent to offspring. DNA and RNA carry genetic information. This genetic information is transferred during cell division, like meiosis or mitosis. The theory of evolution says that all organisms come from the same origin, and College Board wants you to know the evidence that proves organisms share a common ancestry. All organisms use nucleic acids to store and transmit genetic information, and all organisms have ribosomes. If you don't remember what ribosomes are, they make protein. They're an organelle that makes protein. All organisms perform the same metabolic processes. An example of this would be glycolysis is present in both anaerobic and aerobic respiration. Okay, we can finally get into Mendelian genetics. So Gregor Mendel is known as the father of genetics and dedicated his life to experimenting with crossing plants. He also has two laws in his name. The first law is the law of segregation, the separation of alleles. So chromosomes carry alleles, as you should know. When separated into daughter cells during meiosis, these alleles are separated. The separation of alleles allows for genetic variation among gametes. And the second law is the law of independent assortment. Genes are not linked. Two or more genes assort independently of each other. In other words, one gene is not automatically inherited with another gene. For finding the probability of single gene traits from parent to offspring, this formula is used and going to be on the AP Bio formula sheet for the exam, but it's important to understand the formula. To determine probability of A or B happening, you would add the probability of A and the probability of B together. To determine the probability of A and B happening, you would multiply the probabilities together. So we can look at an example. So you can pause the video now if you want to try to do it by yourself, but I'm going to go ahead and explain. Okay, so the first thing you would do is do the crosses. You would do capital B, lowercase b with lowercase b and lowercase b. So you do that cross. And from that cross, you would get 2 brown fur and 2 white fur. And then for the second cross that you would do, which is with lowercase h, and capital H, lowercase h, you would get 2 short fur and 2 long fur. Since this is an and question, because it is asking for an offspring that is brown with long fur, it's an and question, you would multiply. So each of the phenotypes is 1/2. So you would multiply 1/2 times 1/2, and that would give you 1/4. So the answer would be 25%, because 1/4 is equal to 25%. Another thing is you need to make sure you understand how to read pedigrees. College Board is obsessed with this type of chart. If you don't know what a pedigree is, it is a visual representation tracing a trait through a family tree. It helps identify types of inheritance. For example, if a genetic disorder is sex-linked or not. Circles represent females, squares represent males, shaded shapes mean affected, and unshaded shapes mean unaffected. Generations are identified with Roman numerals. Okay, so Unit 5 has a lot of content. I'm going to be making a part 2 with the rest of Unit 5 in it, so stay tuned for that and subscribe and like this video if you learned a lot and it helped you review. Thank you for watching. Bye.